Welcome back, ladies and gentlemen. We're going to continue talking about your cell structure by picking up today talking about your endomembrane system. So I had mentioned to you before about making sure that as you get to this point in your education that you start recognizing what the organelles do, but in the context of the cell types that they would be found in. The other component to this is to keep in mind that none of these organelles are working in isolation from each other. There very much is an interplay between what one organelle is doing and what others are doing in that cell as well. And so we're going to talk today about the endomembrane system because this really is a tight relationship between a set of membranous organelles that drives the way that that cell functions. So being able to see those connections from one organelle to the next is just as important as recognizing what those organelles do individually and why they would be seen in certain concentrations in certain cell types. So when we look at the endomembrane system, the job of the system as a whole is to regulate protein traffic and perform certain metabolic functions. So when we look at the components, we're going to talk about your nucleus and the nuclear envelope, your endoplasmic reticulum, your Golgi apparatus, lysosomes, vacuoles, and the plasma membrane are all sort of a major flow of processes in your cells when we go from one organelle to the next in the system. And these guys are either directly connected to each other, something like the nuclear envelope being directly connected to your ER, or they're connected via the transfer of vesicles, like from the ER to your Golgi or from your Golgi to the plasma membrane. And so, again, these organelles aren't functioning in isolation. So let's take a look at what each of them does individually as we talk through the connections from one to the next. So let's talk your endoplasmic reticulum first. We already covered your nucleus, right? Your nucleus is where your genetic information is, and that genetic information can then get converted into RNA, which leaves the nucleus and likely ends up somewhere in your endoplasmic reticulum. And it is directly continuous with that nuclear envelope. So the membranes of your ER directly connect to the envelope around your nucleus. The endoplasmic reticulum as a whole accounts for about half of the membrane in most eukaryotic cells, which is pretty impressive. It's a pretty large organelle in and of itself, and it's split into two pieces. So you have your rough ER, which is here, and that rough ER is studded by those ribosomes. Remember, those ribosomes are your protein factories. So when we talk about connections from nucleus to ER, well, one of those connections is just the fact that your nucleolus is located in the nucleus, and that's where we produce the ribosomal RNA to build the ribosomes that are attached to that rough ER, but also that flow of information in your cells from your DNA that never leaves that nucleus to creating just those messenger RNA copies of the piece of DNA that I need that day. And that comes out to the rough ER, and that can go to those ribosomes to build the proteins. So transcription happening DNA to RNA here in your nucleus, and then translation happening out here at the ER, specifically on those ribosomes to go from RNA to protein. So your rough ER's general purpose is to be your cell factories, is to be your protein factories in the sense that the ribosomes are attached there to do it. Any proteins that then get produced by that rough ER will end up in transport vesicles, and those will then end up at Golgi that we'll talk about in just a moment. Typically, any of the proteins that get produced here either end up becoming a component of your plasma membrane, so things like glycoproteins, right? Or they end up being secreted from the cell as a whole and used somewhere else in your body. So these guys really are those protein factories for any sort of secreted protein. Your smooth ER kind of always reminds me of like Disney-fied coral. You watched Little Mermaid as a kid. That's kind of what that reminds me of. These guys completely lack ribosomes, and they're much more tubular in their orientation. In fact, if you look at an electron microscopic picture here, you can definitely see the major differences. Um, and your smoothie R can have a diverse set of metabolic functions depending on the type of cell that we find it in. So they can do anything from the synthesis of lipids, additional phospholipids, perhaps for additional membrane building. Um, they can metabolize certain types of carbohydrates, help to break down certain components. Um, and they are also highly concentrated in places like your liver, along with some other organelles, to be part of drug detoxification. So, again, their roles tend to be more dependent on what kind of cell type you find them in. Okay, so let's go back to the idea of protein production. So if I go from my nucleus for my set of instructions, take that ribosomal RNA, I take that messenger RNA, go out to my rough ER, use the ribosome component to then build the protein. That that rough ER will then package that protein into a transport vesicle, and that transport vesicle, more often than not, ends up here, and that's your Golgi apparatus, or you may come across it as your Golgi bodies. So the Golgi apparatus is your center for manufacturing, warehousing, sorting, and shipping. So think of it as a big distribution center if you want an analogy for it. The actual rough ER and the ribosomes are the factories themselves. That product then gets shipped to the distribution center, which is your Golgi apparatus here. So your Golgi apparatus does a handful of things. So it is this sort of flattened membranous sac. It often looks sort of C-shaped because it does have a cis side or a receiving side and a trans side where it will also produce its own vesicles. And the proteins, when they get dropped off here on that receiving side, will end up in these cisternae, these folds, and a handful of things can happen within the Golgi. One of the things that may occur is just some additional manufacturing, additional editing to the proteins. So they may add things to them, they may remove some pieces from them, they may help in adjusting the folding of it. They sort of finalize the product, they clean it up. That's one potential part of what the Golgi do. The other thing that it will do is also determine where that protein is finally going to end up. Is it going to get packaged into something like a lysosome and stay in the cell? Or is it going to end up in another transport vesicle because this is something that needs to be exported from the cell? Or do I need to tag it to stay as part of the cell membrane? So your Golgi apparatus really kind of slaps that shipping label on the product, if you will, and says where it's going to end up. And then it will put those into vesicles on the other side. If it's something like digestive enzymes that are going to stay in the cell, then these vesicles may go off and become lysosomes, or they may end up at the cell membrane and either just get incorporated into the membrane itself if it's something that's meant to stay there, or it can be excreted from the cell if it's meant to be used somewhere else in your body. So speaking of those lysosomes, right, lysosomes are one potential option that those Golgi can end up creating if the protein product is a digestive enzyme or hydrolytic enzymes. You may come across those terms as well, right? To hydrolyze is to break things apart using water. Now, in order for lysosomes to actually perform their ultimate function, they have to merge with a food vacuole. And those food vacuoles are produced when your cells bring in some sort of material. It can be large solid particles, which would be the phagocytosis. So literally translating into cell eating. If they're large liquid particles, then we call it penocytosis. But collectively, this is a process of endocytosis, bringing in larger particles through that cell membrane by creating vacuoles that surround it, bring it in. And then those will merge with those lysosomes in order to digest those components and then those food particles may get dispersed out into the cell for other uses. The other thing that your lysosomes can do is this process called autophagy. So if you have an organelle that's become damaged or just worn out and we need to break it down and get rid of it so I can build new ones, we can also package up those sorts of materials, so internal material, into a vesicle, allow that lysosome to then merge with it and also do the same idea and digest that down into its components, and those components either get removed as waste product or we can turn around and reuse those pieces for something else. Big thing here is that we want to be able to package these things into these membranes because we want to be able to separate those enzymes and keep them contained. If I just let those hydrolytic enzymes free floating in my cytoplasm, they're going to start eating up all sorts of material in my cell, and that's dangerous. We don't want that. You need them compartmentalized so that they're only breaking down what you intend as a cell to break down, as opposed to sort of letting it just break down everything. Okay, so we can talk vacuoles also as part of your endomembrane system. And there's generally, we split them into three major components or three major types. The food vacuoles is what we just talked about, right? Those are the ones that are formed by endocytosis. More often than not, they're specifically phagocytosis, where they're going to store those large food particles that'll merge with those lysosomes and will break that food down. The other two types are contractile vacuoles and central vacuoles. So let's go down to that central vacuole for a second. So central vacuoles are ones that are strictly in your plant cells, and they're usually a pretty large component in the center of that plant cell. So when we look at that, this is going to become important and come back up when we get to chapter 7 and we talk about osmosis, because it is that central vacuole that often is full of water. Now, there's other organic compounds and other things that are also dissolved in there, but we talk about it mostly with the connection to the ability for that water, that osmosis to occur, rush into that central vacuole, and it pushes out it creates pressure, I should say, inside the cell. And that's going to be an important component about why plants can stand up, because plants don't have a skeleton like we do. And so it's going to be that pressure. We'll talk about it as turgor pressure. As that central vacuole fills up, it creates that turgor pressure in the cell. Because plant cells have a cell wall, it allows them to fill up without blowing up. And so we create that pressure, and that gives them that stiffness. If you've ever come across a plant that you haven't watered in a few days, it kind of starts to limp over, and that's because it's losing water. It may not be dead at that point yet, but its central vacuole is shrinking because there's not enough water in it, and so it's not able to maintain its upright position. Contractile vacuoles will also come up when we get to osmosis. And this is a special vacuole that we see in protists. And protists are single-celled eukaryotic animal cells. So these guys don't have that cell wall to be able to prevent themselves from bursting, but they have evolved these things called contractile vacuoles. And if you know what it means to contract, right, we're pumping, all right, we're squeezing. And so these contractile vacuoles will actually increase the number of pumps or contractions every time it gets into a situation where there's too much water rushing in. And so it's basically like a sump pump. If you have one of those in your basement to keep your basement from flooding, that's kind of what the contractile vacuoles are. They'll pump to keep excess water from filling up that cell so that they don't burst, which is pretty cool. Something else I'll mention here, because this is probably something that's new to you, there is a special name for the membrane that surrounds your central vacuole, and that is the thing called a tonoplast. So just in case you come across it, that's probably something you hadn't been taught as an underclassman, so your text brings it up, so I figured I'd bring it up here as well. So when we talk about that tonoplast, it's just a specialized name for the membrane around your central vacuole.